When the muscle cell is in a resting state, the two strands are not in contact with one another. But when your nerves send a message to your muscles, these two protein strands reach out to touch one another. The movement that occurs gives us the name sliding filament theory. In addition to actin and myosin, your muscles also need ATP to provide energy to fuel the sliding movement that occurs when the muscle contracts. It is also important to point out that proteins can change shapes when they come into contact with charged particles called ions. When the muscle is at rest, there are two tiny proteins called tropomyosin and troponin that are wrapped around the actin. There are also ADP molecules stuck to one of the myosin heads when the muscle is in a relaxed state. It is the tropomyosin, troponin, and ADP which keep the myosin heads from extending and attaching to the protected actin strand. During periods of rest, the muscle builds up an abundance of calcium ions that will be used when it is time for the muscles to contract. The cell gets these ions through the calcium pumps of the sarcoplasmic reticulum, which is wrapped around each sarcomere. When the muscle receives a signal from the nerves around it, the signal travels down the cell's membrane in folds called T-tubules. When the signal reaches the SR, the calcium pumps open wide and the calcium ions begin to flow. At this point, the calcium ions bind to the troponin. This causes the troponin to change shape and rotate around the actin, which moves the tropomyosin out of the way, leaving binding sites exposed. When the binding sites open, the myosin heads extend and attach to the actin. This attachment is called a cross bridge. The contact causes the myosin heads to bend towards the center of the sarcomere, shortening the overall length. We call this sarcomere shortening a muscle contraction. When the muscle cell